We're going to talk about PCR, also known as polymerase chain reaction. That's a mouthful. Those are some pretty big words. So let's simplify that, because I'm sure you're asking, what is polymerase chain reaction? Well, it has to do with DNA. And what we're going to do is we're going to take a strand of DNA and we're going to turn it into two identical copies. Okay, so this is complicated, but strangely enough, it only takes about 6 steps in order to copy DNA. But first, we're going to need some ingredients. What we're going to need to do is we're going to need to get a sample DNA. Sampling DNA is not so difficult. It's actually simpler than most people think. A cotton swab can be used to just pull some of the DNA off the skin inside your mouth. And we take that cotton swab, the tip of it now is covered with genetic material, and we take it and we're going to put it inside a test tube. We're going to take that cotton swab and wash it around in there a bit, and that means the DNA is going to come off of the swab. And then it's in the test tube. And we're going to take that. We're going to add something to it. We're going to add some fluid to it. And the fluid we're going to add is a fluid of genetic material. Now, genetic material is just DNA parts. So let's take a moment and talk about what DNA parts are. So DNA has some parts here. These are primers. It's a group of smaller pieces called nucleotides, and these are nucleotides here. So DNA is basically, the fluid is basically primers and nucleotides. Now we take those test tubes full of primers and nucleotides and DNA, and we put them in a steel plate. The steel plate holds a series of test tubes, and that plate then goes onto a very special device. This is a device that can control temperature exactly, and this device is a PCR machine. So all these test tubes sit in that steel plate, and this machine regulates the temperature of what's going to happen in there. So let's move on to step 1, and that's called initialization. So the machine is going to control the temperature, and what it's going to do first is it's going to make the temperature 96 degrees Celsius. And now at 96 degrees Celsius, the DNA is fairly stable. And by that, I mean the rungs between the two sides of the DNA are held together by hydrogen bonds. And at this temperature, they're very stable. Now, we move on to step 2. And in step 2, the machine is going to change the temperature. It's going to raise it. It's going to raise it to 98 degrees Celsius. And this takes us to the stage called denaturation, step 2. So denaturation is where the hydrogen, in a sense, begins to boil and the DNA separates one side from the other. Now remember, this DNA that's now separated is in a fluid that has nucleotides and primers swimming around in the fluid with it. This is important because this takes us to step 3, the annealing phase of the DNA. So the machine, once again, is going to alter our temperature to 60 degrees. It's going to drop down 60 degrees Celsius. Now that drop in temperature is going to make the primers want to adhere to the DNA before it can adhere to itself. So it kind of starts building 2 new chains out of the 2 pieces, and which moves us into step 4, elongation. Elongation is basically kind of a hardening phase. We have a 72 degrees Celsius, and what that's going to do is that these 2 chains are now going to suck up all of the genetic material. It's all going to start bonding together. And Mr. DNA Wizard here, ta-da, we've got 2 chains of DNA. So, now on to step 5, the final elongation. Now this is important. It might seem like these are 2 steps that are very much the same, but they're not. In final elongation, we now change the temperature to 70 degrees Celsius. And what happens here is the DNA is held at this phase for a while. This temperature just happens to kind of bond the DNA a little better. These hydrogen bonds here, it's kind of like our genetic wizard here. He's almost gluing the DNA together. We're getting a tighter bond. Okay. So now, moving on to the final step, we've got 6, final hold. Okay. This is kind of an interesting place here. We drop the temperature drastically to 15 degrees Celsius. Everything in there stays pretty stable. It's almost as if, imagine it's frozen. It's gonna stay exactly how it is at that temperature. We can go ahead and take these copies that we've made and we can stick them in the vault for cold storage, or we can leave them right here inside our magic little machine, the PCR machine, inside that steel plate, if you'll remember, and they can go through another cycle and cycle after cycle after cycle gives us lots and lots of genetic material. And this genetic material can be used, of course, for a series of different scientific purposes. Viola,